Okay, so today we will look at the source code for the perceptron. I don't know how to spell it. So once we understand the individual neuron, how they works, and we can look at this deep neural network framework. We are not going to construct a deep neural network by itself, but we will use MATLAB, the built-in applications, to look at the structure and see how it is run. Then we will look at the source code for LDA analysis. Okay, it's a PCA type of this for discrimination purpose to separate data points belongs to different clusters. Then we will look at a MATLAB app, which provided by MATLAB's classification learner. So this is a MATLAB app. So this is already provided in statistics and the machine learning toolbox with a very nice interface. So for small scale of this classification applications, if you have some source data set from a experiment, and you can convert your Excel spreadsheet data and say, okay, I want to make a small model to predict the outcome. And after that, you have that model, you can use this to predict the next experiment results, for example. Okay, so after that, we will continue to use single cell GA2 to do the annotation, cell annotation, cell type annotation. But this time we use customized database. So marker gene database. As you know, we do have a built-in database, but if you decide I have my own marker gene database, I have some cell type that you are not provided by your current database. How can you customize this and to make this interface work for you, for your subtype of cells? We probably will try single R. This is in R, so call a single R from single cell GA2. See if it will work. Okay. Any questions? Okay. Let's jump into this perceptron framework. So we need input data, which is vector. In order to train this perception, and we need a weight matrix. Okay, so we need two vectors. One is your input data, and another is this weight matrix, weight vector. So in this example, our first data is V1, which is this column. So the outcome we will get is -1. That's our target. So let's see. My first input data, V1, is 1 minus 2, 0, -1. All right. So I'm going to give this to my perceptron that's defined by this vector. So I'm going to use a random 4×1. All right. So I'm going to do a dot product between them. So I can do dot V1 W and ask for the sign. So for the training data number 1, I want to be negative. So that's my goal. Okay, it's working fine for this W setup. I can take a look what's inside W. So this is just for a random number. My second data set, V2, is this column here. Okay, 0, negative 1.5, negative 0.5, negative 1. My expected outcome is still -1. So that's our goal, to set up a parameter in W in order to achieve this. So this time, if I do the sign for second data, it's also good. So it looks like this random guess, this weight is fine, at least for this first 2 input data. So my last training data is the last column, -1, 1, 0.5, -1, transpose. Okay, I check the sign for this last one. So it's still -1, so it's not what we expected. We want to have this, using this training data in order to achieve first data point gave me -1, second gave me -1, the third gave me +1. So at the current setup, my W is not achieve that goal. So I have to check, maybe generate another random number. Another set, I rerun this. Okay, then check the first data, -1, second, -1, third, still -1, so not as way I expected. So you can try this W, and random guess is not a good strategy. So there are better ways to train this, but let's just provide this solution. One of the solution I have is it should have -4.2, -2.8, 1.4. So that's my one of the solution that acceptable for this training purpose. Okay, now if I try the first vector that gave me, W should be 4 numbers. It should be this one. Okay, 0.7, 1.4, 0.7. Okay, so this should be my solution for this small case. There are other solutions, but this should work according to what I know. So this gave you -1. Your second data will be classified into -1. The third will be in +1. Okay. Any questions for this? I have 3 training data, and I gave to this neuron in a series, and for the first data in, second data in, third data. The result, the outcome should be -1, -1, +1. This is my training data because I know the labels for them. Then I change the parameter in my W vector until I achieve this goal. So if I randomly select W into 4 random number, I didn't achieve that. But at a certain stage, when you set up it correctly, you get this -1, -1, +1. Okay. Now you can combine your data, 3 data points into just one vector, one matrix. So V1, V2, V3. Okay, you put the 3 training data in a matrix, so you don't have to run this 3 times. You just V times. So let me make this correct dimension. So W. So that's the matrix for this is 4 by 3, and W is. So I have to transpose my data. Okay, this will make a dot product. And then I will assign function. Okay, so I test my 3 data point input using just one step and multiplicate with vector 1 by 1. So the outcome is -1, -1, 1. All right. So for this solution, so how can I say, okay, my model is trained. Now I use just 3 data points to train this neuron. The solution of the current state of my neuron is 4 numbers. Tells me the model is finished, it's trained. The parameter has been set up. Okay, we finish, we stop training. So now these 4 numbers represent a model, a classification model. I can start to use this to classify my new data point. For instance, if I have a new data point, V4, go to whatever number you can come up with, -0.4, 2, 9, 0. Okay, so I have a new data point. I can test using my current V4, can do dot. So my prediction I made for the new data point, V4, is -1. Okay. So if I say, okay, my V4 is same as V3, so what outcome should be? Because V3, yeah, this should be +1 because I copied the V3, so it's okay. So that's a classification. Okay, so we just use a very small, just single neuron as a model. Now we are going to expand this. We will put a lot of neurons together in parallel, then put in several layers of this and see how can we use this. Yes, just before that, I'm going to show you this function which people use this to learn the parameter, because right now is the one I set it up manually. See, I trained the 3 data points and I found this is the parameters that will give me the best training solution. But if I don't know those numbers, how can I come up with these combinations of parameters? And this function, I believe we do have this in here, the source code. Yeah, so how do we get the 4 parameter values based on your input? This function is here. I have to give this function my input, my output, and some initial random guess about my W. Okay, so what my in the training? So what my training in X? My training X is this V. Okay, I gave 3 input. What my Y for training? Y is the outcome. My Y is -1, -1, and +1. Make sense? So that's what our training data for classification. Okay, so I'm going to give these 2 values, training X and training Y, to this function, to let this function to help me to find the W. So I'm going to give X and Y and some initial random guess of the W. I may have to give the eta. Yeah, so this parameter is for this is a kind of learning parameter, learning rate. So I gave a small number, maybe 0.1, okay? Oh, in the middle, something, the training data need to be transposed probably. Try this, okay? So for this training data, this program and gave me a solution W and in this way, okay? It's different from what I showed you before, manually type that in, okay? So this should work as well. So what I can try is to do some function for this W. So it's gave me -1, -1, +1. So the parameter combinations I found out was using this perception learning this function. Okay, inside here is just try different numbers of combinations in order to achieve the best. Where's the Y value here? So make this small as possible. So some kind of optimization by the iteration. Okay. Yeah. I don't think you have this file, but let me just open this to show you the process again. Yeah. It's a learning rate. If you have iteration, you have to, after finish one step of computation, you have to change your parameter a little bit, larger or small, and then for next iteration to find the solution. So this eta is give you how much difference you want to put into another step. If you have a long jump, you probably will miss around. If you have too small, you may have to learn many steps until you achieve a convergence. So this is called a learning rate. If I change the learning rate, the weight I learned from this changed, because this is a small example, only 3 training data sets. So there are many possible solutions will achieve this. I can share these files later for you, and not essential. The most important is to get the idea, okay? So if I use this training, if I use 0.7, I probably get, yeah, this is all cleaned up. It doesn't matter. So, so the solution looks very different from what I get before. It may be because of this eta, the learning rate is slightly different. But the solution for this small example is this very simple. Okay, so now we are going to look at now. Let's get let's load example data. This is small example data provided by the toolbox of statistics for people to use this as input, which there are 178 different brand, and there are 13 parameters, 13 variables to define those different brand of ones. And then our target is to categorize this 178 into 3 major clusters. Okay, so this from 1 to 60 is the first class. You see, they use three dummy variables to encode these values. So this is group 1, and from 60 to some numbers will be group 2, and then 3 groups. What I'm doing here is just convert this three dummy variable way to indicate the groups and use 1, 2, 3 for classification. So if you look at this y, the output, you will get 1, 2, 3. Just label different variation of this one brand. Can you tell me why people not using this y, this 1, 2, 3, this way to label the one, rather to use like 3 binary 0, 1 to indicate? So when I label the different categories, use numbers 1, 2, 3. That indicate their so this label give you a value for, say, group 3 have a higher value like a 3, the group 1 only 1. So that will make the problem become a regression instead of classification. So if you use label 1, 0, 0 to indicate group 1, 0, 1, 0 indicate group 2, 0, 0, 1 indicate group 3. So you use this so dummy variables, 3 binary variables combination to indicate, to label them. So that's the technique to get rid of this potential regression type of this classification. Make sense? Yeah, so group 1 and 3 it's not because 3 is weighted more, give you 3. It's purely random. So use this label variables to represent the membership ID for them. So let's see. In your MATLAB, do you have this apps, the tab here? Okay, so click this drop down to find show me more. Let's see. Let's just look at this deep neural network design. So I'm not going to design any deep neural network, but just to give you some impression of how this. So because all this operation, you know how to do this one neuron operation. It's just 4 numbers for design one neuron. If you have another, you have another neuron. It's a similar structure, and you have layers. So all this construction computationally, has already become a routine. So people have engineering already designed them. If you can use this graph user interface to set up a neuron, set up 20 neurons, set up multiple layers. So this designer is just give you the way to set up all the parameters and the structures very easy way. Okay, so I click that. I hope the interface will come out. You don't have to do anything, and because underhood, this toolbox already take care for you, and you just define all the structure, how many neurons you want to put in each layers, what activity function in the middle to connect them. Let's see. This is app in my third role. They're machine learning, deep learning. Yeah, let me show. in my third role, machine learning, deep learning, and I click the second app here, the designer. You get it? Maybe it's slow first time run, maybe. Now we can select maybe just this one, and there's some predefined. Okay, we can just click this because it's already installed locally. If you start from scratch, you have to define each layers, but let's just look at a predefined one. So I select this SqueezeNet. Okay, I think this is used for train images. Okay, if you have a picture, you have a label for this picture, say cat, dog. Okay, animal names, and then this network will learn the features and set up parameters. The parameters are within those layers. So orange, the yellow ones are the layers, and orange ones are the activation functions. Okay, so each layers there, I don't know how many neurons in there. So the parameter depends on what's the input data, the matrix, the vectors dimension. Okay, so this deep neural network setup has a input type is image, and for classification, so your training data has to have multiple images, a library, and has labels for each image. The number of layers, there are 68, how to count, 68 layers, and the connections 75. So this overview. We can zoom in so for some of this on top. You start to see some details of those modules. The first one is the image input layer. So you will have input size, you see 227 by 227 and 3 primer colors. So this is like a color picture of the squared as a training input. Then once you have this 3D matrix, three layers of matrix, you put it into a layer called the convolutionary layer of here, and there's some parameters to define this. Okay. Then you gave to activation function. So once you learn the parameter for the from the input data, you do this convolutionary operation. Then you gave you the output to this activity function to allow it to change the values, and then gave to another this pooling 2D layer, pooling size. Where is this? So this is like a pooling layer. I don't know exactly this terminology means, but it just a lot of neurons connected basically. And then another convolutionary 2D layer, another activation function, another 2D layer, and then just keep doing this. There are 68 layers at the end total. It's pretty deep. At the end, you get the classification outcome to put your labeled picture into the correct label bins. Okay. I think how to design this structure, this framework require expertise, and that's beyond the subject of this single cell data analysis. We don't have such a good training data because the picture, the image, and the label is easy to obtain. Their ground truth, their solutions, it's true. But for single cell data, we need a pattern recognition instead of rely on the training. Yeah, some of this maybe you can adapt to the single cell data. So I cannot do a real training because I need to have input data. We need to have a data store, have all these pictures, images, and maybe labeled image from 1 to 1 million. Okay, there are labels for them, cat, dog, horse. Okay. Then after finish the training, you're ready to use this. If you gave it a new picture, it will tell you, okay, this what category or what label in there. Okay, let's look at another neural net pattern recognition. So in this framework, you can design the layers yourself. But here, I believe the structure has been already hard-coded, fixed. And for this purpose, okay, so this is a 2-layer feed-forward network. So you don't have any options to change its structure. So it's already optimized for the problem that you want to solve. Activity function in the middle and how to synthesize this input layers, parameters, and output that gave you a pattern. So let's see we can get some data in. Yeah, so they provide this one data. That's the same data I just load. So we can just import this as an example. So import this. And on your right panel, there is explanation of this training classified predictor set of classes. Okay, the one and the target. So three output. Depends on the parameter combinations of certain variables. Now with this framework, with this input, the 13 variables and the 3 output, we can just start to train this. Yeah. So this 178 data points will be put in, give the neural network one by one, and to achieve this separation of the target 3 class. Okay, finished. So you have training data, all the parameters in the middle through the training process. You can monitor what happened inside in the middle of the training performance. Okay, their validation in the middle test training. The confusion matrix, for example, if you have 3 different clusters of this brand one, then you may correctly, just in the diagonal is 40, 25, 32. So these are the correct assignment class in the training. So in training, they all correctly put in the correct categories. And in test, there's one mistake, which maybe class number 2 has been given to cluster number 3. So you have 1 in this off diagonal position in this confusion matrix. Validation phase, you get 1 mislabeling here. Okay. Overall. Let's see ROC curve for different categories. For 3 categories, and in the test phase, classification 1, cluster 1 is always 100% true positive and 0 false negative. So this training is really work really good for cluster 1, class 1, this blue curve. There's some mistake for class 2 and 3. So ROC curve is not perfect. Otherwise, they're perfect. So in the training phase, it's perfect. Okay. So in order to test, we have to give new data points to do the test. We don't have a new data, so it's okay. We just leave it here. So you get the idea is enough. You can write this out as a function. So this function will take new variables, new data points, and give you a prediction. Any questions for this neural network stuff? Okay. So let's see, we are done with these 2, finished. So I do have this LDA source code to share with you. So this is a PCA-like type of this transposition. Is rotate your data in order to allow them to, from this angle, to separate them best. Let's see. I can just quickly run this as a illustration. So I will clear all run section, yes. And then I load a data set. So this is from iris data, is some flowers, I believe. And then you have your label. You have your input data. So let's see, input data is 150 species, subspecies of some flower. And then you have parameters 1, 2, 3, 4 to describe each of this sample. Then you have a label, group 1, group 2, group 3, basically. Now you give the LDA function. Okay. So I'm just run this. This function return to me this rotation variable, rotation matrix. Okay. Remember, rotation matrix, it's a squared matrix. In this case, it's 4 by 4 because the data points in the 4D space. So we need this rotation matrix to rotate your data. So I'm going to plot this and see what's going on. I think this is like 4 different variables. If you look at their values in original, so those are the 150 samples. Each is a values for those samples. After rotation, you create the first PC, can separate 3 categories very clearly. Okay, otherwise you have to sometimes you cannot separate them, sometimes you can, but once you rotate your data to this new angle, and then your first PC just clearly separate 3 classes. Okay. So I will share with you this source code. If you have interesting, you can go back and play with later. So I'm going to during the break, I will drop this zipped file, and it contains both LDA test and also this perception code for. Okay. Let's take a 5 minutes break. I just drag this into the shared folder. Now we are going to look at another built-in app. This again, not for single cell, it's for general purpose of classification. The application is called Classification Learner. So you can type this. Yes, just before that, let me create a test data set so we can have some variables to play with, okay? So I clear my space and then load patients. So this is a built-in data set. I'm going to define, so that's how many patients. We have 100 patients. I will define the input. So as age, you can use a tab. You should have this because the variable name already here, so you should be automatically complete by after using the tab. Systolic. Heart. Remember, it's a case sensitive, so the capital H, systolic. Weight. Okay, I finish my definition of this data table. I want to use these parameters to predict the gender. Okay, so I can have Y gender. Okay, so this is a classification problem. So give you age, diastolic height, systolic weight. Can you predict the gender from this given data set? Okay, so we are going to use this classification learner. Okay. So this is a comprehensive collection of a lot of non-neural network classification algorithm put under the same interface. It will try all these combinations. You can use Bayesian inference. You can use LDA. We have, yeah, what's the question? Oh, this is optional. So those are the column variables. You can put a comma. You can leave it like that. It's fine. Okay, so this is interface. Do you have this right now on your computer? Everybody get it? It's good. So you can check. So so this program is located in machine learning apps, okay, under this folder. So it's M function basically. We can check what other programs in there. So if I do dir and put, so there are other regression learner here, the classification. Yeah, there are two, there's only two under this folder. So the one we just start is this. Okay, now we can start a new session and use variables from the workspace we already prepared for ourselves with this input output. Otherwise, you may provide external file, which I believe should be in certain format. You have a Y and X. So the classification problem is to use X predict Y. Okay. From the workspace, we can load input data, which is big X, okay, contains 5 variables. Response is also from the workspace, is our Y. Okay. So within X, we have 5 parameters. Remember there. So these 5 parameters are in the column 1, 2, 5. And the first column, remember their age. So we have a range is from 25 to 50. Another is systolic blood pressure. I can't remember anyway. So this gave you the range, the preview. If you know, you can include or exclude, for example, age. It's not related to the gender. So you may, if you have the impression that's not important, you can deselect it. Otherwise, you keep it. Okay, so I will leave it like checked. And then here you select cross-validation fold. In the class, I talk about the 10-fold CV, and you can see like a 5-fold or more. Okay, so once you finish, we can just start to train the model. Okay, so their scatter plot to show the combinations of 2 variables. In this column 1 against column 2. And this labeled male, female. We know column 1 is really not any relevant in here, so the age. We may want to look at column 4, column 3. Column 2 and 3 combinations seems have some separation power. Column 4 and 2 is okay, not good. Yeah, it looks like column 5 is really useful variable. if you plus 3 and 5, maybe give you better separation. Anyway, so this algorithm will use these combinations of those variables to use decision tree, LDA, logistic regression, naive Bayesian, support vector. So basically tried everything if you allow it to do it. So I can say, okay, quick to all train, okay. So it will run selected algorithm to help you to fit the data, okay. Or you can select, I really want to use Gaussian naive Bayesian, and you can select this. And do the training. The result will be on this panel, in this panel. So after you finished, you can take a look of the results. And especially you know what the confusion matrix is. And then maybe use ROC curve to validate. And you can try multiple different algorithms. For this simple example, any algorithm should work very well. So it doesn't matter. But for some more complicated situation, maybe one classifier is stand out from others. So you probably want to keep that. So model 1 I'm using here is Bayesian, naive Bayesian. The number model 2 is logistic regression. It all achieve this accuracy 100%. So no mislabel. We can also look at the confusion matrix. This is a confusion matrix. We can also look at ROC curve. Again, this should be perfect. Model 1 is also perfect if you use ROC curve. Okay. Is that clear for you? Then you are ready to test your data. So if you have new data comes in, you can read this into a variable, put it in the workspace, and let it to predict. Okay. Or you can still select the simple neural network classifiers at the end here. For some training job, you probably want to switch this parallel, use parallel to accelerate the computation, because when you switch this on, some training job will distribute it to the multiple cores in your computer to take advantage of this parallel computing. Okay, we are okay with this. No questions. So I'm going to turn off, switch off this. Yeah. Connection between genes. So it's a gene network. I mentioned from the last lecture. I don't remember which part. Okay. Yeah. So we will have a lecture specifically discuss gene-gene interaction network. Yeah. But for this random forest per se, it's not for constructing gene-gene. You can use this as a way to construct gene regulatory network. You have to translate your problem to build a gene-gene interaction network into a classification problem, because random forest is an algorithm for classification. Good question. So we will discuss network construction. So I can clean my desktop here and ready to use. So this function allow you to change your directory, basically the CD to GA, okay, to the toolbox itself. What I want to show you here is where the database is located, the database of marker genes for annotation. So within this folder, so you install this toolbox here, and within this folder, there's the run class, third party, yeah. So under this, there is Bangalore DB database is located here. So we will take a look of this database. Found it? Found the folder? Okay. No, not yet. Okay, have to make sure you have this. Not input. You don't have the toolbox installed, so you have to search for. Yeah. Maybe there's a new computer. You was there, so that's a new computer for you. So if you don't have this, you have to go back to download. Okay, you have to install it. Yeah. So I want to find this folder so we can locate the database for the annotation. Yeah. It's not here. No, within the run. Okay. Class run. Yeah. Down. internal, third party. Oh, external, maybe. Third party. Oh, third party. Yes, so it's arranged differently because you are using, yeah. Do you have that folder? No? Okay, it's, yeah, so that's inside, yes. The current folder, you click the home button, then the layout, check default, the first one. Then you should have this, yeah. Yeah, so because you don't have this add parse, maybe not working for you, but it's fine. It's fine, yeah. Yeah, so installed. Then you just click here inside, and then you run. Right, yes. Third party and DB, yeah. The first one, yes, yeah. So this folder is where I save this BanglaDB database. Yeah. So you know, if you visit that website, BanglaDB, you can download the Excel spreadsheet. Yes, that's within here. Yes, that's right. Yeah. Okay, so looks like everybody has already found the folder. Yes. Oh. Now you can double click this folder and find those files. Okay. All the information you need is saved in this so-called marker MAT. You can double click this. HS is for human, right? MM is for mouse, and DR is for zebrafish. Okay, so when you double click, you will load the two variables into your workspace. These are just two tables. The first one is a list contain 181 variable one, cell type, and their marker genes. So the information I got is directly from the website. And just reformalized and put in here. Okay, put in a table which MATLAB can read. That's where the annotation, and we'll look at those marker genes expression and give you the most likely ones on the top. Okay. But as you said, as you can see, there are limitations for this. For example, some genes maybe some cell type maybe you are interested in not in there, so you'll never be able to have a correct annotation for the cell you really want to start it. So you have to find a way to expand this, right? If you want to have your cell type. Another table is a two-column table. Is gene. So those genes are all the genes appeared in this table and their weight. I mentioned the weight means whether they are highly specific to some certain marker cell type. If this gene appeared in all cell types, they are not very informative. So the weight of that gene will be 1. Otherwise, will be higher. So each gene have a slightly different contributions for this specificity of cell type annotation. So okay, now so this file can be easily read by MATLAB, but not a human. So what you can do is to open this file, marker list, and open. So this is easy to read. It's the same table, but right in the text format. Okay. In the middle of cell type and the marker gene, there's a tab. It's invisible from here, but just make sure you know, okay, there's a tab here to separate these two columns. And the rest of the marker genes is just a long list of genes that's separated by the comma. This database is from BangaloreDB. Let me find is this website. It's this. So you can design your own if you have better collection or there's one database you just want to use for yourself, it's fine. You can customize it. So I'm going to show you once you have your customized database, how can you annotate cells? Okay. For instance, I just copy some of this from here as an example. Okay. So for instance, I have my own let me make a new edit plus notepad. I can just use notepad. For instance, I have my own, this is my own data. One, two, three. I have my mini database for myself. Now let's load the example data. Raw data, for example, completely new. Okay, we start from scratch. You know, you can do embedding first. So at this step, I just prepared my own database for classification, for annotation. Now you know you can cluster cells into 20 clusters. Now you can annotate using the built-in database, but if you don't want, try experiment. I'm not so sure today we can make it, but if there's a bug, I will go back and fix. Okay, so what you can do is to select from the menu from annotation cell type using customized markers. Okay, so click this. Now go back to this one you just prepared for this purpose. Copy it, replace this one. Okay, so I'm not so sure today I will make it, but I just give it a try. So the annotation will be cell type 1, cell type 2. Yeah, so the function hasn't been complete, but I will do that later. What you can get is something like this, a table. We have 20 clusters. It depends on the value. So cluster S1 will be given ID, the cell type as a 3, because the value is largest. Cluster 2 will be 40, will be cell type 3. Cluster 3 will be cell type 5. Okay, so this function, it works, but not completely annotation automatically. So I will continue to work on this function. But once I have this table, I can easily to label those cells based on this cell type name you provided. Okay, so next time we will have this function. But you get the idea, all right? I think this function is useful when you have your own definition of subcell types. So you extract neurons, for example, then you can have your subtype of neurons definition, then you focus on neurons and to separate them into subclusters. No, this will automatically help you to build the weight and everything automatically. You provide the table. Yeah. Yeah, right now you can do that in this way. Next week I will improve these functions. We will automatically label them. Yeah. I know the function is there, but I just don't have time to finish it. Mm-hmm. Okay. I think the last thing today I want to try is to see whether this external function single R is package in R, can run on this PC correctly. So I'm going to just give it a try. It's crashes and crashes. That's, I don't know. So let's see. So if you want to view what's going on, this the data matrix will send it to R, and R will run give annotation, and then we will read the back. This is mouse data. Yeah, if you look at the back end, oh, it's stopped. Something wrong. Install package. This arrow, yes. So install package into our. Yeah, so we don't have a single R package installed. I don't know how to install it. So in this case, I'm going to search and install single R in my R environment here. So where's my R? RStudio. We need to find a way to install singular R. I hope after installation we can run. Okay, so that's what I found from Google install single R. By the way, I don't think we need it. I don't believe single R will give you a better annotation at all. But it just show you the way you probably want to try. Yeah.
We copy paste this one into.
Yes, in R, not MATLAB. I say R. Yeah, yeah. I don't want to upload. So I should have library single R. Yeah, looks like it's fine. I can install it. So I hope this time I'm fine. It's already. You can check the back end here. Well, it's still installing something. So those are the R output. I don't know what's going on there. It's trying to install some dependence library. I think it's still working on that, this R package. I just wait and see whether the communication between two programs will happen. Where we are waiting, I probably want to find the article to show you how slow R is. So the comparison R versus Julia. Okay, so this is a blog. By a professor in California Department of Public Health. He's our user for many years and transferred to Julia. And here he compared how long it will take in R to compute this summation from 1 to this 1 billion. And in R, it takes this many seconds to achieve. Well, for Julia, it takes only 150 milliseconds. Okay, so it's 70,000 times slower in this kind of operation. In MATLAB, it takes 1.2 seconds for the same operation. But this, of course, is a very special case. Okay, my finished. I don't know the annotation. Label cell type. So I believe this single R gave me the answer like this. Is that what we expected? I don't think so. I remember I have a lot of different cell types. So this, to me, single R is a complete crap. Let me load my pre-processed data and see what our annotation is. Maybe I use it in a different way, made it wrong. So that's our own annotation. We do have a T cell. We have beta cell here. I don't